I think we'll start. I'm Roger Morris, I'm head of school. The traditional role of the head of school, in this case of biomedical sciences, is to say something of not great relevance while everyone comes into the lecture theatre and then the true lecturer takes over. So first off, I would like very warmly to welcome everyone here. You are the widest array of audience I think we've ever had. We have chemistry teachers from school, and an entire class of students and various individual students. We have King's students ourselves, we have alumni all over the place, and we have the Royal Society of Chemistry and chemists primarily from London, but from the south of England really, who are all here to help us celebrate the return of chemistry to King's. I particularly would like at the very beginning to say thanks both to the alumni and to the chemistry departments and the Royal Society of Chemistry for giving us support in getting started, ongoing. Everyone is a pleasure to work with and thrilled to see chemistry back at King's and help us make sure, as it definitely is, back to stay. Okay, so since chemistry was closed in 2003, up to about 40 chemists have been employed at King's to work in medical imaging, and Professor Phil Blower will be taking over from me in a moment, and certainly he and the others in medical imaging have been a major driving force to the return of chemistry, but also in drug delivery and discovery, in structural biology, in material science. Chemists are there in all these places pursuing their discipline in particular applications. And from them came the demand that we have a real chemistry department to champion chemistry per se. And it's one which we all agree with anyway, because chemistry, the science of the making and breaking of bonds, working from principles rather than empirical observations, is the chemistry. It's a science of change, and the science of change is the science of life and the universe and all of that. We have missed chemistry. We can't do without it. We're delighted to have it back. And we're delighted to have you here to celebrate it with us. Now, in keeping the demand for chemistry alive, a key figure has actually been Chris Dobson, who, from a point when people said, well, is it possible to get it back in some form or other, said, look, you have a blank slate. You have a new opportunity to get new chemists to reinvent chemistry or reinvent how they apply chemistry, to rethink it, and to run a new course where you think it from the beginning. And that has very much turned out to be the case. But before getting on to that, you've seen photos around this particular lump of old wood I'm grasping. This actually is here because the chemistry at King's has, well, the physical sciences at King's have always been closely related to the biological sciences. What I have here in my hands is Michael Faraday's lectern, from which he gave his lectures at the Royal Society, which he left to Sir William Gull, who was the governor of Guy's Hospital, and with whom he did experiments on electricity in living tissue. Life was more exciting before the NHS came in if you got into hospital. And since then, a large amount of good chemistry and physics at King's has involved its integration with biological problems, as Ricky Eckert will touch on briefly in a moment. So Chris said we were going to get new and exciting generation of scientists, and that we advertised. We were looking for 6 posts, and we certainly have. And you're going to get an array or description of their work and a few samples in a moment. We are continuing that investment, and amongst the continued investment, we're now looking for a new head to lead this exciting department. I raise that point now. Why miss a chance? Okay. So as you get to know our current chemists in chemistry, keep that in mind, and it's my pleasure now to hand over to Professor Phil Blower. So I'll say again, Phil has been a major driver in getting this department restarted, including having been acting head for the last 12 months He was a young man 12 months ago. He was somewhat changed with the stresses and strains of the job, but I'm sure he'll be delighted to take over now. Thank you, Phil.
Ricky's got the unenviable task of talking about the research of the department in general, other people's research. It's a tricky thing to do. Ricky joined us, what, a year and a half, 2 years ago from Harvard, and actually she still makes a few trips across the pond to Harvard in order to complete the research that she has been doing now, so she'll be with us full-time soon. But Ricky's going to introduce the research profile of the department. All yours.
Hello, can everybody hear me? Okay, so as Phil said, I will tell you about the research in the department. I'll give you an introduction to our research and just a very brief overview. And so I'll just give you a little bit about information about the background and the environment of the department, and then I'll tell you about the individual staff members' work. And so, as was pointed out in the beginning, the department consists of a growing number of core chemists and a large number of chemists who work in other divisions in other schools who are chemists or have some element of their research in chemistry, but are embedded in other divisions. And we're quite keen to preserve that structure in this new department because we feel it really helps us to maintain the very interdisciplinary nature of our work, and we're very keen to do that. And so you can see here after on this little chart here that shows chemistry really at the center of the interdisciplinary, different types of interdisciplinary work. So we work with scientists in physics, we work with scientists in the School of Medicine, and in various different applications and different departments in biomedical sciences. The department has recently moved into a new building, as some of you already also saw in your booklet. It's a building that used to belong to a biotech company, and we've taken it over recently, which means that it has very nice lab space set up both for chemistry and for biology. At present, there are also other groups who have overlapping interests in this building. And it's a quite close walk to both the Guy's campus and the Waterloo campus at which we're here right now. So we already heard the historical example of Michael Faraday as one of the luminaries of science associated with King's. And I'll just give you a couple of other historical examples, although a little bit more recent than Michael Faraday, about the kind of work that has been going on at King's for a very long time at the interface between the physical sciences and biology research. And so you all know, or you may have noticed, that we're actually in the Franklin Wilkins building. And this Franklin Wilkins building is named after Rosalind Franklin and Maurice Wilkins, who were instrumental in discovering the structure of DNA. You can see here this famous photo of 51, which showed that DNA is a helix, was taken by Rosalind Franklin here at King's. And then Maurice Wilkins, together with Watson and Crick, was awarded the Nobel Prize for the discovery. Rosalind Franklin had already died at a very young age at that point, unfortunately. I'll show you one other example of a historical figure associated with King's. And I chose this example because this really exemplifies how one can move between disciplines and really make major contributions to science and to health. And this example is James Black. So he's moved throughout his career. He moved between academia and industry. And he really made major insights into drug discovery because he was one of the pioneers in terms of actually understanding biological mechanism and then designing drugs to target this biological mechanism in a systematic and rational way. And by doing so, he discovered major drug classes, including the beta blockers, which are still used in the clinic to treat coronary heart disease, and also different drugs that are used to treat ulcers. A little bit more modern now is that King's has a very long tradition and is very strong in analytical and forensic science. And you can see this example just from last year here, that the Drug Control Center here was actually used together with GSK, were involved in doing all the doping testings for the Olympic and Paralympic Games. Okay, so this just gives you a flavor for the kind of environment that the department has started in. And now I'll tell you about the research in the department. And so I'll give you an overview. As I said, research in the department uses techniques, approaches, and ideas from chemistry to study a broad range of problems ranging from physics to chemistry and biology. And so that's why I'm using this little cartoon here to illustrate our work. We go from understanding single chemical bonds to whole animals. And at this point, I'd like to thank all my colleagues for giving me slides that I'll show you during this talk and for patiently explaining to me what those slides actually show. And I apologize if I mangle our slides. So what I've done here is I've grouped our research into different themes, and I'll go through these themes and explain what people are working on. Okay, so our first theme is computational and physical chemistry and biophysics. And so as we just saw from the example of Rosalind Franklin, traditionally biophysics has been very strong here, with many researchers in the Randall Division of Cell and Molecular Biophysics, in which I also have an appointment in addition to chemistry. And so many of our affiliated chemistry department members also have appointments in physics and in the Randall Division. And then, as you'll see in the next couple of slides, many of the new staff that we've hired are very strong in computational and physical chemistry. And so the first staff member you see here is Maria Sands. She's a physical chemist who uses rotational spectroscopy to study the structure of biomolecules, and she will give a talk describing her research in a little bit more detail right after me, so I won't tell you anything more about her work because she can do it much better herself. So then we have, I'll tell you about the work of two of our computational chemists. This is Carmen Domene, and maybe I should try and dim the lights a little bit again. Baby? Okay. So now you can see Carmen in her full glory here. So Carmen is a computational chemist, and she's interested in studying a wide range of different biological problems that we'll come to on the next slide. And the motivation for Carmen's research is that she wants to use her computational approaches to help interpret biological data, and to also see if, by doing the modeling that she does, one can avoid having to do complicated and expensive experiments. And maybe this work will actually lead to the prediction of new properties for the systems that she's interested in. So she uses a number of different computational approaches, ranging from coarse-grained modeling to molecular dynamics and these QMM methods, which we'll hear a little bit more about when I talk about the research of my next colleague. And so what the different topics that Carmen is interested in is that she has a main focus on studying membrane protein biophysics. How do membranes and proteins interact with each other? And she's done some very nice work in this area on ion channels and transporters, where she's interested in gating and conductance. She's also more recently gotten interested in studying anesthetics, because with many of these anesthetics, one doesn't know, do they bind to proteins, do they bind to membranes? And so she's using her computational approaches to predict function of these different molecules, these different anesthetic molecules. And then at a slightly larger level, if you want to call that, not at the molecular level, she's also very interested in how proteins and membranes interact with each other during viral fusion. For example, the example that she's focused on is in HIV viral entry. In a different area, she's interested in understanding how proteins recognize the DNA binding, areas that the proteins bind to on DNA. She's interested in modeling that. And in a long ongoing project in collaboration with Chris Schofield at Oxford, she's interested in understanding how human oxygen sensing works. This is a complex process where many different reactions occur, and she's modeling many of the different bond-making and breaking reactions and different charge transfers. She's also interested in neutron analysis tools, and this was a new one for me, where basically this is a technique that combines theoretical and experimental approaches, where one can experimentally label a molecule with deuterium, for example, and she's doing this with her anesthetics. And then the experimentalist takes these molecules to the neutron reactor that will then give you data about where this anesthetic actually binds, so it gives her information, does it bind to proteins, does it bind to the membrane. Okay, so this is Carmen's work. Our other computational chemist is Adina Rasta, and she also uses computational approaches, but her main interest is in understanding the role of phosphates in biology. And so you all know that phosphates are really important in many different biological processes. They're important in DNA, in RNA, so in the storage and transfer of genetic materials. They're important in signaling, and they're important in energy storage. And many of the reactions that involve the transfer of phosphate in proteins are very dynamic, and so it's quite difficult to use experimental methods to study this. And so Adina uses computational approaches to model what happens exactly at the enzyme active site. And the approaches, she uses a range of approaches, as I just described for Carmen, ranging from the accurate ab initio quantum mechanics methods, which are very accurate but are also very time-consuming, to some broader approaches. And she especially uses this approach here that I just mentioned already, the quantum mechanics, molecular mechanics is a hybrid. And so she uses the accurate quantum mechanics to model the enzyme active site and the slightly less, easier to compute molecular mechanics to study the remainder of the protein. And so the project that she's interested in is she's interested in ERK kinase, in the dynamics of RNA polymerase, and in the metal ion catalyzed NTP hydrolysis. And I'll just show you one example of some of the work that she's done in a little bit more detail. So Adina wanted to understand how does this enzyme here called RNase H, which is endonuclease, it cleaves RNA, how does the mechanism of this cleavage occur? And you can see this cartoon here. So the protein in blue binds to the RNA in red, and you cleave this bond here. And so it wasn't possible to determine this experimentally, and so the modeling that Adina used then resulted in understanding how this mechanism works, which is that there's a coupled proton transfer, that the proton transfer is coupled to the cleavage of this phosphate backbone bond. And she developed a new computational approach to do this, which is called the Hamiltonian replica exchange coupled string method. And this is a way in which one can measure bond breaking and bond making simultaneously. Okay, our last researcher in the physical sciences category is Sergey Nazarov, and I'm not very good at pronouncing Spanish names, which is a problem because we have many Spanish people in our department. So sorry, I didn't say it quite right. But so Sergey is in the Department of Physics and in the Randall Division, and he's an affiliate member of the chemistry department. And so one of the main questions that Sergey is interested in his lab is how are mechanical forces in nature transmitted? And the folding and unfolding of proteins is a very important question in biology, and we just heard a lot from Chris about how different proteins fold and misfold and form aggregations. They can also be under quite a lot of mechanical stress, for example, in muscles. As the muscle moves, the proteins within it also have to move. And so Sergey is interested in understanding the energetics of the folding of these types of proteins. And you can see here is a little cartoon of a protein. And if one wants to study how it unfolds using what people normally do when they study protein folding, which is a chemical denaturation, then the size of the protein actually doesn't change very much. But if you pull on it and mechanically unfold it, the size of the protein changes a lot, so that really gives you an idea of the of the energetic landscape associated with the protein folding. And Sergey studies this using single molecule force clamp spectroscopy, and we heard a little bit about a similar technique just before. Basically, the way this works is that you have a chain of polyproteins, and they're trapped between this piezoelectric, what's it called? I can't read the last bit, and the cantilever, and you can pull on this, and then you can measure the forces associated with changes in folding. And there's just a little animation that shows this again. So this shows in more detail. The cantilever is attached to the chain of proteins here, and as this unfolds, we can measure the change in energy. Okay. And so what I just told you was about the unfolding of proteins, but how proteins fold is also important, and it's a very complicated energy surface of how these proteins fold. There are many different possibilities on how it could happen. And so Sergey uses, again, a mechanical folding of his force clamp experiments to study this question. And so this is a trace that shows what the experiment looks like. So if you pull on your chain of proteins here, you unfold it, then you release the folding. You can watch the protein refold. And then this is very unique for this type of experimental approach because then you can pull on it again and you can see if it's folded properly and what the energetics of this folding process involved. And so this is the folding and unfolding of proteins and measuring the energy. So the energy landscape of a chemical reaction is much simpler, as we can see here. And this allows us to actually use this approach to study the energetics of a specific chemical bond. And the specific chemical bond here is the formation of this disulfide bond here. And so the experimental setup that Sergey uses is, again, the same experiment here, but instead of just pulling on the protein, this disulfide bond is attached here, and it needs a nucleophile to cleave it. So once the protein is completely stretched, he adds a nucleophile, he watches the change in energy, and that gives us information about the energy of the specific chemical reaction. And that's what this trace shows here. So we see the changes in energy as the protein unfolds, and then we see the energy associated with the cleavage of the different disulfide bonds. So that's all I'll tell you about our physical chemistry and biophysics group, Computational. Then we have a very large group of people working in the general area of chemical biology and drug discovery. And this is because the traditional strengths of King's are in biomedicine, and so many of the people who do chemistry and also the people who recently joined the department have some interest in biological applications of their chemistry. And so, there are many different groups that have different types of chemical and chemical biology backgrounds, and there are also some spin-out companies that have come out of the research that was done here. And so I've grouped these, I've divided this team into two themes. And if you ask a chemical biologist for the definition of chemical biology, you probably get as many definitions as there are people that you ask. But I think two of the main definitions are to either study biology at a chemical level, and that's what this theme is, or to use chemical tools to study biology, and that's what our next theme is. So this theme is chemical and structural biology, where researchers are studying proteins, their mechanisms and assembly at the chemical level. And a number of our departmental affiliates are structural biologists. And they're very lucky to have this Center of Biomedical Spectroscopy that was set up with a grant from the Wellcome Trust. And it really is another interdisciplinary center that sits between basic and clinical science. And they have very nice NMRs and all these other different spectroscopy methods to study the biology that they're interested in. And so our example here is Rivka Isaacson. She's a structural biologist mostly. And she's interested in applying different approaches to understand how biological mechanisms work. And the example that's shown here is from proteins that are called, what are they called? Tail-anchored proteins, sorry. So many things to remember. Tail-anchored proteins are they're really important proteins in many different types of families. They have different jobs, and they're different from other transmembrane proteins because they don't get inserted into membranes the same way. So there has to be a completely different machinery that's dedicated to inserting these tail-anchored proteins into membranes. And this is the machinery that Rivka is interested in studying. It's called the GET pathway for tail-anchored protein insertion. And she's interested in the first step of this pathway. And that's what this protein is shown here. She solved the structure of this protein. She purified it. She crystallized it. She solved the structure of the protein. And then using crystallography, and then by NMR, she solved how this protein interacts with its binding partner in this very first step of the machinery that inserts these tail-anchored proteins. And so now that she has this static information of the structure of this protein, she wants to know how do these different proteins of the pathway interact with each other. And for that, she uses some more dynamic approaches. So this is basically similar to the structure that we saw on the previous slide, but this gives us some dynamic information because the red tells us the regions of this complex that can move, that can be dynamic. And this was determined using 3 different approaches. This microscale thermophoresis, which is a new piece of equipment that we've recently gotten and many people are quite excited about because it can give you information about the affinity and stoichiometry of different proteins binding to each other. She also used isothermal titration calorimetry. Down here you can see NMR experiments where she uses labeled nitrogen, and you can see how these peaks here shift, and that gives you information about how dynamic this complex is. So Brendan Orner is interested in molecular recognition, and he has a number of different projects that get towards this. And so the first project here is that Brendan wants to understand the energetics of protein cage assembly, and he uses this to make nanoparticles. And so there are 2 goals, really. One is to understand better how different proteins can interact with each other, and the other is to make these nanoparticles that have very well-defined biophysical properties. And so the way it's done is you have these ferritin molecules here, and they can be made to assemble into different types of protein cages, and then these little gold particles go on the inside of these cages, and that's what gives you the nanoparticles. And then, using different types of tethering mechanisms, and one is shown here, that can have different symmetry. They can be tetrahedral or octahedral. You can then assemble these little particles and cages into many different types of arrays, and can test to see how these different, what the properties are of these arrays that have different symmetries and different sizes. So in another project, he's interested in using a combinatorial synthesis, in a way, to develop catalysts that can catalyze different chemical reactions. And the reactions that he's the most interested in are, for this project, are bioorthogonal reactions. So that means that they contain, that the reaction can work in the same environment as an aqueous environment, in the cellular environment, but does not contain any functional groups that are found in cells. And I can't see it here, but the functional groups that he uses for this are, for example, azides and aldehydes. And so he basically uses approaches from molecular biology to make a library of different cyclic peptides that will then catalyze this reaction. And the assay is that he has a different fluorophore attached to each of the 2 reaction partners, and when they meet, he gets a fluorescent signal. And the last project is to try and make mimetics of carbohydrates. And we'll hear a little bit more about carbohydrates further along, but basically many different carbohydrates are displayed on cell surfaces, and they vary depending on what type of cell exists. And so if one can bind to those selectively, then one can actually look to see how the surface of the cell changes, for example, during differentiation or development. And so he has a number of different assays that allow one to make different patterns of how the carbohydrates on the cell surface. Okay, so the second theme within our chemical biology group is the small molecules in chemical biology. And so there are 2 themes in here. One is that we study chemicals that naturally occur in biology, and these are metabolites or other kinds of small molecules that cells make for some kind of reason. And then there are many people who use chemistry to make tools to study biology. And this can be either to develop small molecule tools, or in the Institute of Pharmaceutical Science, there are a number of researchers who work on nanomaterials for biological applications, and we just saw an example of nanomaterials in Brendan's work. And so Sarah Barry is interested in small molecules that are made by different bacteria, and we'll hear about her work in more detail at the end of the session. And then we've got Gad Wagner. He's a chemical biologist and a medicinal chemist, and he's interested in developing small molecule inhibitors of enzymes that are hard to target. And the main group of enzymes he's interested in are the glycosyltransferases, although he's interested in some other enzymes as well. And the motivation for this is really that there are many classes of enzymes where it would be very interesting to have chemical tools to study these enzymes, and also for drug development, but they just haven't been targeted by the pharmaceutical industry because they're considered difficult. And so this is really a very nice contribution that we can make in academia, is to study these enzymes and to show that they actually can be targeted. And so the glycosyltransferases are one of these families. They're really important because they make lots of different structures in cells that have many different roles. For example, I just told you about how they are displayed on the cell surface. And what these enzymes do is they basically just link two sugars to each other to make different chains of sugars. And we'll see an example of this on the next slide. Another family of enzymes that GET is also interested in are these NADP-dependent enzymes that are involved in, they're non-classical redox enzymes that are involved in post-translational covalent modification of different types of proteins. But here's just a little bit more detail about the glycosyltransferase inhibitors. So the goal is to control cellular glycosylation with small molecules. And so what's the use of having small molecules that can inhibit glycosyltransferases? We can learn more about the enzymology of these different enzymes because we know quite little about them relative to many other enzyme families. GET can also try to control the synthesis of different sugars on the cell surface. And this is what one of the things that this cartoon here illustrates, is that if we have an inhibitor here, we can see that the carbohydrates displayed on the cell surface are different, and then one can study the function of changing the carbohydrates there. And one can also use them these inhibitors, once he's developed them, for chemical biology and drug discovery in these specific projects that involve specific enzymes that are important in glycosyltransferase. Okay, and this is me. So my lab is interested in using chemical approaches to study cell division. And so these slides are the easiest for me to explain, I hope. So we're really interested in trying to apply our chemical approaches to study cell division. And why do we do that? Because it's an important basic biological process about which quite little is actually known. But you cannot have life growth or development without cell division. It's also very dynamic. Many things happen very quickly, and if you have small molecules that act within seconds that you can add to cells, and you can freeze different stages of cell division, then you can study what's happening at those specific stages. And so it'd be very nice to have small molecules that inhibit cell division. And they've been used to great effect in terms of understanding how the shape of cells is defined and how cells divide. And before we started this work, and we're mostly focused on cytokinesis, which is the physical division of cells, there just weren't that many small molecules available to do this. So it would be very useful to have more of them. It's also important in terms of potential chemotherapy, because many of you might know that Taxol is one of the main cancer drugs that's used to treat an array of different cancers, especially breast cancer, and that targets mitosis. So it's quite clinically relevant. But basically, the overall philosophy of my lab is that I believe that chemical biology is really a key tool, not the only tool, but a key tool in the systematic analysis of complex biology. And so we have a number of different research programs to address these questions that I just mentioned. So we've done a number of screens to develop and discover different small molecule inhibitors of cytokinesis. One specific project where we've done that is that we're interested in the role of Rho signaling during cytokinesis. So Rho GTPase is our key regulator of the cell division process. And we use a number of different tools to understand how these different proteins in the pathway participate in this process. We've also more recently gotten interested in trying to understand how signals from the outside might influence the cell division machinery. And one of those ways in which that could happen are through G-protein coupled receptors. They're important signaling proteins. And we're also interested in how these signals are then trafficked through cells during the division process. And then lastly, we've just recently completed a study to understand the role of what I mentioned earlier, naturally occurring small molecules, the role of lipids in the process of cell division. The plasma membrane and many other lipid compartments hugely change as a cell divides, and nobody knew anything about the chemical nature of the lipids involved. And which is really amazing because cells make tens of thousands of different lipids, and we know very little about these different lipids. And so we've done a study using different approaches, including mass spectrometry and cell biology, to really figure out how the lipids change as the cell goes through the cell cycle and where these lipids are localized. Okay, and this brings me to our last theme, is imaging chemistry. So imaging chemistry is led by Phil Blower, and it's based at St. Thomas's Hospital. And they have all these different approaches, MRI, X-ray, PET spec, and ultrasound. And they can do it in small animals, they can do it in people, and they really have a big focus on the rapid translation of their application into the clinic. Okay, so this is Phil. You've already met him. And so Phil has a large number of projects, and that's why I'm showing this table here of his different projects to just give you a flavor, and then I'll show you one project in a little more detail afterwards. So he uses all these different radionuclides to incorporate them into different types of probes for imaging. He can incorporate into all these different things, proteins, peptides, nanoparticles, and so on. And these are relevant to all these many important diseases. Okay, and so this is the example that shows the imaging of a tumor in a head and neck cancer. So this is the tumor, I think. And this is the imaging complex that's used here. So it has copper at its center, and copper can work as a positron emitter, and so that's what's actually imaged here. And so Phil's group uses inorganic chemistry and organic chemistry to make these different ligands that then bind to the copper, and then once the ligand gets taken up or diffuses into the cell, if the cell has the right redox environment, as it does in the tumor here, the ligand is cleaved and the copper is trapped in the cell, and that's what emits, and that's what allows you to visualize these tumors here. So that's what I just said. So that's really the range of the type of work that's done in Phil's lab. Okay, and this is the last colleague whose work I'll tell you about here. This is Tom Eakin. He's also in imaging and an affiliate member of the chemistry department. And Tom is interested in using NMR applications in biomeds, and you can see his NMR machine here with his hyperpolarized NMR machine next to it. And he can really look at the range going from the animal, this is a transverse image of a rat, to looking at which metabolites are present in a perfused organ or in an animal. And then he uses this hyperpolarized NMR, which increases the signal of the C13 carbon, so it allows you to look at different metabolites that contain carbons, and you can use that to study their kinetics in perfused organs or even in little animals. This is an example of some of the work that Tom has done. This is an NMR trace of tissues that were taken from either a transgenic animal or a control animal. A transgenic animal has a mutation in a protein involved in cardiovascular disease. And by analyzing these NMR traces and looking at what kind of metabolites exist, you can do principal component analysis and really show that these transgenic and control samples are in quite different, have quite different metabolisms. And I chose this as the last example in this talk because I think it really kind of takes us full circle. So we started talking about bonds and molecules to proteins, to cells, to animals, and now we're back again to talking about individual molecules. And for me, this was also a lesson that communication in science is actually quite important, because as I was talking to all my colleagues about their research, I didn't know that Tom was working on this, and now we've decided that we would actually collaborate on this work with his work and our work on lipids, because it's a very nice complement. So there's always something that can be learned in science if you talk to your colleagues. Okay, so thank you very much to all of our funders who fund all the work in all the groups that we have in our department here. And as you've seen, we have many young groups in this department from all over the world who do a wide range of different chemical sciences. And we're very excited to be here now, and we're very excited to interact with you, the chemical community, further. So thank you. Okay, so I'm going to focus
talk about biomolecules and what is the information that we can get when we study them using rotational spectroscopy. So we heard a little bit of biomolecules today. As you know, biomolecules are any molecules that occur in living organisms, and they can range from single molecules, such as amino acids, nucleic acid bases, to oligopeptides and oligosaccharides, to big macromolecules, such as the proteins we have seen today, DNA, RNA, etc. So the molecules we focused on in our work are the single molecules, but the fact that they are single doesn't mean that they're simple. Biomolecules have a large functional flexibility. They have a few, or quite a few single bonds, and of course those single bonds around which the atoms can rotate give rise to a large number of low-energy conformations, as we can see, for example, with the amino acid isoleucine. So we have these small biomolecules that present a huge number of conformations. What is the structure that this biomolecule is going to adopt? How can we know about this? So if we want to know about this, we have to look at the forces that take place within the biomolecule, the intramolecular forces, mainly hydrogen bonds that take place between the functional groups in the molecule, and also if the biomolecule is within a solvent, for example, we have to look at the intermolecular forces. And the structure that the biomolecule is going to adopt depends on the relative importance of these forces. If we want to know about the structures of these biomolecules, then we have to study these forces. And the way of doing that is to study biomolecules in the gas phase, because then is when we can study the molecules isolated and we can get the intrinsic molecular properties. If we study the biomolecules in the condensed phase, we also get the effects of the medium. And it's very difficult to disentangle them from the molecular properties. So by studying biomolecules in the gas phase, we can get to know their conformational preferences and also which are the intramolecular interactions that are more relevant. And once we know about the bare biomolecules, then we can go on to study them interacting with solvents like water or with other partners, like, for example, what you would find in receptors in cells. And then we can get to know about the intermolecular interactions. The other reason why we want to study biomolecules in the gas phase is because the dielectric constant of vacuum is closer to the dielectric constant that we can find in active sites in proteins and membrane interiors than that in bulk water. So in fact, we can get the results of this obtained in the gas phase as good models to know about the behavior of biomolecules in proteins. So how are we going to study these biomolecules? We use rotational spectroscopy. So we apply radiation in the microwave region of the electromagnetic spectrum. And what happens is that we make the molecules change the way in which they rotate. And when they change how they rotate, they emit certain signals that we collect in the rotational spectrum, and then we analyze them, and we can obtain the structure. And the beauty of this technique is that it's very sensitive, it has very high resolution, so we can identify very clearly different conformations of the same biomolecule, such as, for example, these two conformers of the amino acid alanine that you can see have completely different spectrum, and they are also down to the disposition of this, the configuration of this amino group, and also the configuration of the carboxylic group. So we can take the rotational spectra as fingerprints for the biomolecular structure. So with this technique, we have investigated different types of biomolecules, amino acid, nucleic acid bases, neurotransmitters, and also we've studied microsolvation, which is what happens when you surround the biomolecule just by a few molecules of the solvent, in this case water. And this is what I'm going to tell you about today, our microsolvation studies. And the reason why we wanted to investigate microsolvation, and in particular in amino acids, is because in the gas phase, amino acids are neutral molecules, while in solution, they are doubly charged ions. We have a deprotonated carboxylic group and a protonated amino group. So, and very little is known about how this happens. I mean, we don't know how many water molecules we need to pass from this neutral state to the switcher ion form of an amino acid. We don't know exactly where the water molecule binds, And also, one of the important questions is what happens with the structure of the biomolecule when we go from having the biomolecule isolated to when it's interacting with water or other solvents. Does it change? Is the conformation that is preferred in the isolated biomolecule the same as when it's interacting with water, or is it different? So to do this, we started investigating the interaction of glycine with one water molecule, because glycine is the simplest amino acid, so this is the simplest molecular model to study amino acid-water interactions, and also this is the first step of hydration. For glycine, we have several conformations, as you can see here, for the bare amino acid. So we consider all of them when we thought about what we can find as the interaction or the interacting glycine with water. And we also consider the different positions or places where water can interact with the different conformations. So can interact with this functional group, with both, or depending on the conformer it's interacting with, bind, for example, to this carboxylic group or to these other two functional groups in that conformation. So we consider all of this, and then we went into analyzing the rotational spectrum, and we found just signals that were arising from one structure of this complex. And this was the structure that we found, where water is binding to the carboxylic group of glycine through two different hydrogen bonds. Glycine is still in its neutral form, and also the water molecule is not perturbing these intramolecular interactions of glycine that are stabilizing the bare conformation. In fact, the conformation of glycine in the complex is the same as when it is isolated. So we observed this for the interaction of glycine with one water molecule. What happens if we add more water molecules? And we also determined the structure. I forgot to say that. This is another information that we can get from rotational spectroscopy. So then we investigated the complex with two water molecules, and we found that the two waters interact with the carboxylic group of glycine, forming this 8-membered train. And they are both, as in the former case, behaving as proton donors and acceptors. Glycine is still in its neutral form and is still maintaining the same conformation as it had when it was isolated. So then we thought, okay, so what about if we change the amino acid? So we started with a very simple change, which is remove one of the hydrogens of glycine and we attach a methyl group. So we studied alanine with water, with 1 and 2 water molecules, and what we found was that the water molecule, again, attaches to the carboxylic group, just in the case of the complexes with 1 water molecule, and also when we have 2 water molecules. Alanine, as glycine, is still in its neutral form and maintains the same conformation as it had when it was isolated. So in fact, the change from glycine to alanine is a smaller structural change, and it doesn't seem to affect the microsolvation process at these levels of microsolvation, that is interacting only with 1 and 2 water molecules. So of course now we would like to study microsolvation in different amino acids, in polar amino acids, where we have other functional groups and we have a lot more conformational variety, to know if water can affect them more, if it induces more somatic changes. And also, we would like to investigate other biologically relevant molecules, such as odorants. And the reason for this is because the molecular mechanisms of smell are not well understood. And one of the intriguing things is that we have molecules that are enantiomers, that have the same structure, are mirror images of each other, but like our hands, they're not superimposable. And these pairs of enantiomers, there are some of them that smell the same, and some of them that smell differently. And this has been attributed very recently to conformational flexibility. So we would like to know, studying prototypical systems, whether this is true, if conformational flexibility affects how we perceive these molecules, and also if there are any changes if we make them interact with mimics of smell receptors. So I hope that I gave you an idea of the information that can be extracted using rotational spectroscopy from biomolecules. And I would like to thank my student, Miguel Ángel Rodríguez. Also, most of the work that I've shown here today has been done in my previous university in Spain, so I'd like to thank all the people in the molecular spectroscopy group there, and also Dr. Bravo from Germany, because of his contribution to the computer. Thank you.
University of Warwick, and works at the interface, I suppose you could say, between chemistry and microbiology, I suppose. So, enough filling in time. You've got it on the screen. All yours, Sarah.
Okay. Oh, there we go. That works. So I hope what I'm going to tell you in the next 10 minutes is a bit of a sprint through natural products and why they're important, why their biosynthesis and studying their biosynthesis is important, and a little bit about what I've done in this area and what I hope to do in the future. So a number of you, I'm sure, in this audience will be familiar with some of the compounds that are on this slide. And these are highly complex molecules, and they're all natural products produced by microorganisms, so either bacteria or fungi. And we can divide them into two groups. Those in black are the good natural products, those that we use to treat a variety of different diseases or conditions. And those in red are natural products, and I guess the bad natural products, those that are produced by microorganisms as virulence factors in a disease. So they're important in the causes of disease. And we can divide them into two groups as well based on their biosynthesis. Those in blue are non-ribosomal peptides, and those in black are polyketides. And these are two of the major families of microbial natural products. Now, I'm going to focus on non-ribosomal peptides just to give you an idea of how these things are chemically put together in cells. And I hope what you'll see, first of all, is the chemical complexity and diversity of these molecules. And secondly, that these are no ordinary peptides. And we've already seen ribosomally produced peptides, which ultimately go on to produce proteins. But these are highly modified peptides, often involving amino acids that are non-proteinogenic, so none of the 20 amino acids that we make proteins from. And you'll also see some very exotic functionality in some of these compounds. And the question is how, if the biosynthesis of these compounds is often controlled largely by highly conserved machinery, how do we introduce this chemical complexity into the molecules? What are the enzymes involved in doing that? The reasons that, or one of the reasons that I guess are interested in studying it is the mechanisms behind, the chemical mechanisms behind those enzymes are interesting, because synthetically often these reactions are either very challenging or impossible currently. So if I run through how non-ribosomal peptides are biosynthesized in bacteria or fungi, and this is a very schematic version of what a non-ribosomal peptide might look like. And this is entirely hypothetical non-ribosomal peptide synthetase. And to give you an idea of what the protein itself might look like, so if we take a module, and explain what that is in a moment, what you have is the 4 different domains in this module. So each circle represents an enzymatic domain, so it has different activity. And these are very large proteins, so an individual module has a size of about 150 to 200 kilodaltons. And you imagine this is a single polypeptide, then you've got a very, very large protein. And you can have 4 modules, but it could also be 10 modules or 2 modules. So they vary from between different pathways. So basically, non-ribosomal peptide synthetases work like little pharmaceutical factories, if you like. They're assembly lines. And they build up the peptide step by step, and each module is responsible for the addition of a new amino acid. And in the course of this, the peptide is always bound to the enzyme. And the peptidal carrier protein, the PCP domain, essentially acts like a conveyor belt in this factory. The adenylation domain acts as a selection tool, so it selects each individual amino acid. It's always, in each domain, it's specific for a single amino acid. And the condensation domain does the chemistry, so it forms the peptide bonds that make up the peptide. So in that context, the chemistry itself, in this case, is not particularly exciting. We're just making peptide bonds. So if we want to introduce some interesting functionality or different kinds of functional groups, one of the ways that bacteria do this is by using non-proteinogenic amino acids. And so in the gene cluster that will harbor the gene that encodes this protein, there will also be genes that encode other enzymes that biosynthesize different amino acids or carry out reactions later on in the biosynthetic pathway. So if we take a notional enzyme and we generate a new amino acid from proteinogenic amino acid, and our adenylation domain then is selective for this amino acid, and it catalyzes the covalent attachment of this amino acid to the carrier protein. And we do that for each of the other modules. And now we are set up to basically make our tetrapeptide. And so we do some amide bond formation, and we end up with a tetrapeptide. So it should be obvious that if you have 4 modules, you get a tetrapeptide. This is a linear process. If you have 10 modules, you get a decapeptide, and so on. So now we need to cleave off our natural product from the enzyme, and we do that using a nucleophile. So this could be water, it could be an internal hydroxyl group or an amine, in which case you would get a cyclic peptide. So we're going to generate a linear peptide in this case. And now we can go on to decorate that with further functionality and make it even more complex. So in this case, what we call these enzymes or these reactions are tailoring reactions. We could start introducing hydroxyl groups, methyl groups, but there are numerous different variations from halogenases to dehydratases and so on. So the possibilities are, in theory, endless. But what we should remember is that each of these tailoring enzymes are very selective. They are pathway specific, so they will only act in this pathway and not another pathway within the same microorganism. And that's what makes them all the more interesting, because they are highly evolved to be extremely specific to often very complex molecules. And sometimes they evolve chemistry that is, well, almost unique to specific pathways. And that's what makes them interesting to study. So I'm going to talk a little bit more about some tailoring enzymes that I've worked on. So this is work that I did in the University of Warwick under Professor Greg Chalice. And we were working on an organism known as Streptomyces scabies, which is an unusual Streptomyces species in that it's pathogenic, it's plant pathogenic. And this pathogen works by making a deceptively simple cyclic dipeptide, and this is non-ribosomally produced, and its biological activity hinges on this nitro group. And these compounds are interesting because nitrated natural products are quite rare. We were interested as to how this nitro group was biosynthetically introduced. So our collaborators in Cornell University, they had identified the gene cluster on which all of the biosynthetic genes that were required to make this compound were located. So one of the advantages of studying microbial genetics, well, particularly bacterial genetics, is when you're looking for these kinds of biosynthetic gene clusters, they are often located nicely together on the genome. And they basically found that these two enzymes, which are non-ribosomal peptide synthetases, are involved in making the dipeptide from these two amino acid precursors. This cytochrome P450 likely catalyzes hydroxylation at 2 positions on the dipeptide to give you fractamin A, which is the final compound. And then they found that TXTD and TXTE, which are a nitric oxide synthase and cytochrome P450, were probably involved in making this very unusual amino acid precursor. But genetic experiments only got them to this point. And they hypothesized, they found initially that TXTD, the nitric oxide synthase, was only involved in generating nitric oxide from arginine. So it acts like a normal nitric oxide synthase. And so the question was, what was this other cytochrome P450 doing, and how, if it was involved in doing this nitration reaction, how was it doing it? And so we came in at this point to try and understand some of the biochemistry involved in this system. So we cloned TXTE and overexpressed it in E. coli. We purified the protein, and this is a normal P450. So all P450s contain heme chrome 4, which is ligated to the protein by a highly conserved cysteine residue. And this is the kind of standard UV visible assay for cytochrome P450. So you reduce the iron to iron 2 and you bind carbon monoxide and you get this characteristic band. So we found that tryptophan could indeed bind to this enzyme, so we can use UV visible spectroscopy to generate a binding curve. This interaction, as you might expect, is stereospecific. So D-tryptophan does not bind to the enzyme, and nor does a range of other aliphatic amino acids. So we suspected that this might be a substrate for TXTE, so we incubated it in vitro with nitric oxide in the presence of electron transport proteins, and NADPH is an electron source. And when you do the reaction, in the control reaction where you've inactivated this protein, you get no change, but in the test reaction you can see this nice yellow color developing. And so we use LCMS to analyze the reactions, and in the test reaction you can see there's a new peak, and that peak has a mass that corresponds to a nitrated tryptophan. What this doesn't tell you is where that nitro group is located on the aromatic ring. And so in order to find that out, we scale up this reaction. When I say scale up, we scale it up to 50 micrograms. And we isolate enough of the material and purify it by HPLC, and that allows us to compare it to the synthetic standard. And you can see, all I'm showing here is the aromatic region of the NMR spectrum. You can see the chemical shifts are identical for all of the protons, and so are the coupling constants, or the coupling pattern. So that tells you that there is only a single regioisomer present. So in the compound, it is only the 4-nitrotryptophan that we observe, and we don't observe any other regioisomers. And we also don't observe any other hydroxylation or epoxidation products, any other oxidation chemistry going on here. So this enzyme is extremely unusual. It has evolved a function as a P450 that is completely, to this point, unknown for this superfamily of enzymes. And a very specific function as well. It's highly selective in that it nitrates only at this position on tryptophan. And this is interesting from a chemist's point of view, because doing this synthetically, i.e. nitrating an aromatic ring using a nitrating agent and gaining only the 4-nitro product would be exceptionally difficult. And so we study these kinds of enzymes to see if we can either get inspiration, I suppose, for generating new catalysts for synthetic use, or as possibly biocatalysts in themselves. So we've studied a lot of the enzymes in this pathway. So I've already mentioned TXTE. I'm currently working on TXTD, which is a nitric oxide synthase, and again, highly unusual because it's the only nitric oxide synthase known to be involved in a biosynthetic pathway. And we've also studied TXTC and shown its activity, and that should be published shortly. So my current work is basically in a similar area. We're interested in identifying new natural products, and hopefully those that might be useful therapeutically, but also in the biosynthesis of pathways of virulence factors that might be involved in disease. In elucidating the orders of steps of these biosynthetic pathways, and identifying new reactions, identifying interesting and exciting chemistry to study, because the mechanisms themselves are interesting and also, again, potentially very useful. And also, finally, understanding the molecular mechanisms of these things is not, well, it's interesting academically, but also if we think about the complex compounds that I showed you in the first slide, synthetically they're exceptionally difficult to make. If what you have is bacteria, which are little mini synthetic chemists, it would be preferable to get these things to make these molecules for you, and also to make derivatives. And the only way that we can do that is by genetically manipulating these organisms in order to make things that we would like, so the rational design of biosynthetic pathways. And that's the ultimate goal of many of people who work in this area, but the only way that we can hope to get to that point is to understand the molecular mechanisms involved in these things. And when you take such a very, very large protein, that's a huge amount of work, and very little is really understood about how these things function. So I guess I'll leave it there. There's a number of people I'd like to thank. So Greg was my supervisor in Warwick and really introduced me to this whole area. These are a number of students that work in the Axterman project, our collaborators in Cornell and in Manchester who worked out who helped us work out the TXTE mechanism, which if anyone is interested in, I'm happy to talk about. These are funders on that project. These are my current funders. And I have to thank you for your attention, and also for coming here today. And because Ricky asked me to, tea and coffee is available now on the first floor upstairs. And I'm glad some of you at least recognize what that is. Thank you very much.
Thank you for the introduction. This is correct. I'm 50% in chemistry now, but when I joined the college just under 3 years ago, I was initially 100% in pharmaceutical science, and I still am 50% in pharmaceutical science now. I'm the program director for the new undergraduate program, Chemistry with Biomedicine, and I've been involved in the development almost since the beginning of the program. And what I want to do in the next 20 minutes or so is give you an idea of where we are with the program, where we want to go, but also where we came from. And sometimes it feels like when we started out, that time is a very, very long time ago indeed, and that it's been a very long journey. It's been an interesting journey, but actually it's been a rather short, if very intense journey. So we started with the development of the program in spring 2011. We formed a steering group to develop the program. We then had to complete a lot of forms and submit that to external review and internal review. This was approved about 2 years ago, and then over the past 12 months we recruited a significant number of staff, and in September 2012 took in our first group of students, and they have just sat the exams. And had it not been for the fact that I talked to one of the students before this meeting, I would have given a lot away, which I'm not supposed to give away because the students don't know the results of the exams yet. But what I can tell you is if there are students in the audience, most of you don't have to worry too much. So my own personal journey started with this email from Roger, sent on the 24th of January in 2011. I was surprised I still had the email, but I probably kept it for a reason. It was sent to quite a small number of people and had a very clear instruction to get going on relaunching an undergraduate chemistry program. It also tells you something interesting, well, first about the timelines, and then also about the way Roger motivates his staff and gets the best out of people by saying that whatever he put together in this document was way out of his expertise, but it will annoy you so much that you will suggest much better alternatives. Now, I don't know if this is true, but looking at the old document again, which I have to say, the program as it is now still very much embodies the vision you put down at the very beginning. So for better or worse, and I think for much, much better, really the course as it is now is very much, to a large extent, your brainchild. And at the time when we started, of course, there was a bit of a chemistry-shaped hole at the college because there wasn't a chemistry department as such, which is not the same as saying, as we've already seen a number of times today, that there wasn't any chemistry science going on. Indeed, even after the closure of the old chemistry department, chemistry as a science, as a subject, continued to flourish at the department, at the college, just in other departments, like nutritional science, imaging, pharmaceutical science in particular, the Drug Control Center. We've seen examples for all of this. So now you can look at this, obviously, and moan and complain, or you can really see it as an opportunity. And I think this is what staff very high up in the college, as Chris said, recognize that this is really an opportunity. This is an opportunity to bring in a new undergraduate program which teaches science at the molecular level, and that, of course, is what chemistry is, but also interfaces with these applied aspects of chemistry in these existing departments and divisions. And that would then be, of course, a two-way street. The students that would emerge from such a course would be perfectly placed to move on to, say, postgraduate degrees or research positions in these departments here or elsewhere, but also because we already have this corollary of chemistry and chemistry-oriented research and scientists at the college. These people could contribute to the teaching, the development, and the teaching, the delivery of the course. That was the idea, and I'm going to tell you to what degree we managed to implement this. So why chemistry with biomedicine? I guess there were two reasons from my perspective. One, obviously, King's traditionally has been very strong in medicine, in biology, and in biomedicine, so there was an existing framework there. But then second, also chemistry and biomedicine are or represent an obvious and very well linked up pair. If you take a clinical scientist, a medical doctor, say, who's interested in lung diseases, asthma, say, then obviously he would be interested in what happens to the organ as such. If you're a cell biologist, you might be interested in what happens at the cellular level in the lung when cells interact with each other. If you are a pharmacologist, then maybe you look one level closer at the interaction of particular molecules at the cell surface with particular receptors. But of course, if you really want to understand what's going on here, what you're talking about is chemistry. Because you're talking about what's happening at the molecular level, you're talking about biomolecules, as we've heard about, you're talking about molecular interactions. So if you really want to make a contribution to biomedical problems, then I think the chemistry approach of doing this is a very valuable and a very important one, because chemistry ultimately underpins many aspects of these other disciplines. So why did we think we could pull this off? Again, I think that there are a number of reasons. First, there was a very clear and very strong commitment from a managerial level at the college, and I have to say, certainly from my perspective, all of these commitments have been honored to this day, and we saw one example with the recent acquisition of the new physical home of chemistry at Britannia House. A second reason, and this is not trivial, is that we had a chemistry teaching laboratory. And I say this is not trivial because Phil Blower and myself went to an RSC meeting not too long ago with representatives from a number of institutions across the UK which are starting or in the process of starting new chemistry degree programs. So we're not the only ones. I would like to think of us as some sort of avant-garde, but we're not the only ones. But many of these other institutions, simply in terms of the infrastructure, have to literally start from scratch, and we didn't. We had a very good infrastructure, so we have very good infrastructure, and that includes the teaching lab in this building here. And then thirdly, of course, we had a very large group of people who could really sit down and start drafting the program on a pretty, admittedly, on a pretty tight time scale. And this is as good a time as any, I guess, to really thank everyone who has contributed to the development of the program and who got us to where we are today. So I'm sure I forgot a few names. I'm very sorry about this. I just want to mention two people explicitly. One, I mentioned Roger, of course, already, so without Roger we wouldn't be here today, but also without Phil we wouldn't be here today, who spent an enormous amount of time, had an enormous input in the development of the program, over and above his role of leading the imaging science department. And then I also want to mention one person who has since retired. Alison Mainwood was a professor in physics, and she helped us a lot, especially in the very early stages of the program, where the activity was really sitting down, finding the right forms, and completing the right forms in the correct way, and Alison has been a great help in that regard. So there's a very large number of people who did a lot of work early on to get things off the ground. And then over the past year, of course, we gradually recruited a pretty significant number of new staff members who have all then been very heavily involved in the further development and the delivery of the course. And I really just want to mention one person in particular. We've already heard about Maria's research, and she's a very good example for what is expected of staff here and what tremendous achievements staff have delivered. So she moved from Spain about a year ago, so she's obviously setting up a research group here. In addition to that, she's made a significant contribution to the teaching of physical chemistry. She's acting as the admissions tutor. I'm very glad they've taken over that role from me. And she's also looking after our outreach program. So my day usually has around 24 hours, but I think Maria has found a way to extend that to about 28 or 32 hours. But it's, of course, not just her, like really everyone in this group of people. On the academic side, we have a teaching fellow, Alice, the technicians, Mel and her teacher, and then on the managerial and admin side, Rebecca and Will has done a fantastic job over the past year to really make the vision we had at the beginning work in practice. And then, of course, from the start, we worked with the RSC to make sure we're really developing this as a chemistry program because it was designed as a chemistry program, so that we don't end up after 4 years having something which is maybe not the chemistry program, though nice it may be, because we're working, of course, also towards accreditation by the RSC. So we sat down with them, and David and Toby are in the audience to begin with, and ask for advice, sort of what role chemistry would have to play in such an interdisciplinary type of program. I was actually quite surprised, to be honest. The advice was relatively, or the attitude is relatively hands-off. There were really two main pieces of advice we received. Make clear that the core chemistry elements are recognizable. So if you have a physical chemistry module, call it physical chemistry, so that the reviewers, the examiners, recognize this as physical chemistry. But if you have lots of biology in the module, then we're not too worked up about that. And then the other aspect really is to give the students a sufficient amount of practical laboratory experience, which is, of course, another hallmark of a chemistry program, as it should be. So our idea then, in very general terms, was to bring together three main important aspects of chemistry: the synthesis of making things, as Roger already alluded to, an important discriminator, for example, compared to subjects like biochemistry, the ability, the techniques, and the intellectual understanding to analyze things like molecules, atoms, ions, and so forth, and then also have a big element where the students get experience or being taught in applying their knowledge in specific applications in the biomedical arena. This is what the course structure looks like. So we've just come to the end of the first year. So in black you can see the core elements of chemistry. We place a great emphasis on maths, and we really benefited here from an existing module in physics, which helped us get the whole program off the ground. But all the other three modules and the laboratory were developed from scratch by us in the department. And we're going to move on to year 2. You can see the program is front-loaded with chemistry, a lot of black here, even here as well. The biomedical elements then come in or are being introduced gradually as the students move through the program. And you also see that in year 1 and year 2 we only have one mega laboratory, if you want to call it that, where the individual laboratory, the individual experiment brings together, or is designed to bring together, the different strands of chemistry and also elements of biology, biochemistry, and biomedicine. In years 3 and 4, the students will then be moving towards applications. We also introduce an element of choice where they can choose an individual optional module. We have a laboratory module in year 3 where the students also will be led closer towards methods, methodology, and techniques used in research, and that, of course, will stand them in good stead in the final year, where they will carry out a 6-month research project which complements a range of different modules, many of which already exist at the college in the area of biomedicine. So I want to say a few more words coming to the end of the talk about some of the things we've already done, where we already know whether or not they have worked, and I'm only going to talk about the things which have worked. So the laboratory in the first year, which was to a large extent designed and organized by Alice, our teaching fellow, comprises 20 experiments in total, which gives the students about 160 hours in the lab, and with that we're on track with regard to the RSC expectations. And the individual experiments, as I said, are designed in a very integrated fashion. This is one example, the very last experiment. The students synthesized, did some inorganic chemistry, synthesized some metal complexes, and then used these as catalysts in an organic reaction. So we want to build in from the start this idea that you're not just making a molecule and then we're going to put it in the fridge or we're going to pour it down in the sink. You actually do chemistry to do some further chemistry or biology or analytical work with it. This is what some of the students have produced, although, to be honest, Alice, I think it's your samples, is that right? Anyway, so the student samples look a bit like that as well, so these are 3 of the inorganic catalysts they made. They then use them for the organic transformation, and this is what a typical year 1 student write-up looks like. So you will not be surprised to hear that we're actually quite pleased with the quality of our students. It's a small group of students, but a very select group of students. The achievements in the lab wouldn't have been possible without the support of the lab staff, and that's not just support from the academics, but in particular also from Alice, Hatice, and Mel. And at the moment, at least, we have an excellent staff-student ratio, certainly in the laboratory. So if there are students in the audience who are looking for, I wouldn't quite say a one-to-one tuition, but a very good supervision in the laboratory, we certainly at the moment can offer that. And then we're also using this expertise and the repertoire of experiments we've already built up and our facilities to work with the community. This is one example. Towards the tail end of last year, we got involved in an outreach activity with the St. Paul's School in London, where St. Paul's actually worked together with excellent students from state schools to give them practical laboratory experience in chemistry. And the first 4 sessions they did at St. Paul's School, and then the final session we did here, because of course equipment like an NMR machine or even an IR spectrophotometer is not normally available at a secondary school, and we were very happy to provide these instruments for a Saturday morning experience. And we're also participating in the University of London Taster Days. We've run one of those only very recently, a few weeks ago. This is the program where the students really go, have a few hours experience what life at the university is like, both in terms of lectures or tutorial style teaching, and then of course also in terms of the, laboratory sessions, which are usually I guess the biggest hit of the day. And for that session I want to thank in particular Alice, Maria, Sarah, Rivka, Mel and Hatice, and keep an eye on these names. If you look at the laboratory session, as I said already, that was a very big hit with the students. And if you look at these pictures, and if you think about the names on the previous slide, and if I think about one of Stuart's slides, I think chemistry very clearly has a future, and that future very clearly is female. And we like the Taster Days, the students also like the Taster Days. And this was one of the, shall I say, typical types of responses we got from this student, who obviously had a very good day indeed. So are we there yet? Of course we're not there yet. We've just finished the first year. We've completed a number of important milestones, but there are equally important milestones ahead of us. Obviously the next academic year, both in terms of planning, preparation, and then execution, will be important. Then years 3 and 4, ultimately, of course, the graduation of our first cohort of students, and then hopefully at that point in time, the accreditation through the RSC. So I'm not quite sure whether we're still here. We're probably somewhere around here. We've got a few more bends to go around, but it's a very bright future we're moving towards, I'm sure. Thank you.
With characteristic modesty, I think you can see that while Gerd mentioned everybody else, he failed to mention himself, and the amount of energy he's put into it. I was particularly impressed that you managed to thank Alison Maywood, even though she's no longer here. She was a professor in physics, was critical to getting it going, and I think it's lovely that you should mention it. Equally, as you can see, Gerd is in himself a rare beast in this chemistry department, since he is clearly male. Yes, the question at the back.
I have listened your presentation, and I was very surprised that you probably, I suppose, never worked in pharmaceutical industry. So you have put your emphasis on bioproducts, did you?
Sorry, emphasis on?
Bioproducts, biomedicines.
Well, that was the name we came up with. I actually wasn't involved in that very first decision, I have to say. I understand that there have been long and complex discussions down to the level whether it should be, the title of the course should be chemistry with biomedicine or chemistry and biomedicine. I guess what the course should be, and what hopefully is captured in the title, is that it's a chemistry course. So that's really very important. It is the chemistry program, at the heart of it, is the teaching and the learning of chemistry. But this is embedded in specific applications of chemistry, most of which are in the biological or biomedical arena. And this is how we arrived at that title.
Okay.
We're talking to partners in industry. We want to offer the students the opportunity in the third and in particular the fourth year to do placements in industry. So we're certainly not under the impression that we know sort of exactly what's going on in the real world. But I would maybe in that sense take the word biomedicine with a pinch of salt.
Okay, I agree with you, because biomedicine has particular trend. It's going actually in production of organic drugs, which could be produced on different ways. So that's very important to distinguish what you are actually going to teach your students. In regard to your applied chemistry, I would say, in pharmaceutical industry or food industry or whatever, there are a lot of chemists who work there. And from my 10 years experience in research in pharmaceutical industry and quality control, I think you missed the point. Because what these students need to know are synthesis, unfortunately, because we still rely on synthesis, how they are made, physical chemistry.
We do need to move on. Perhaps we can talk to Gerd afterwards rather than consuming the whole room. Fortunately, we're on a very tight time schedule.
Okay.
So I'm sure Gerd would be happy to talk to you afterwards. It gives me great pleasure to introduce Hagen, who is the Professor of Chemical Biology from Oxford. And he has interests in protein chemistry, in molecular genetics. And over to you, Hagen.
All right, thank you.
I'd like to introduce the principal of the college. If I could introduce Professor Sergey Nazarov. Thank you. For those of you who don't know, trying to walk down these stairs regularly is terrible, because they're about a step and a half and you can't quite work out.
Well, I'm conscious that I and my colleague, Vice Principal Evelyn Welch, who's going to speak to you in a minute, that we stand between you and the party. So this is a heavy responsibility. But I hope very much that you've had a good day. To judge from what I've heard of the last presentation, you have had a very good day. I wish I could have been here for more of it. I have a good excuse for why I wasn't here, because I was taking a prominent part in medical school graduations, which are not to be trifled with. So I spent the day in Southwark Cathedral with several hundred new medical graduates. But I did want to take this opportunity, even at the cost of keeping you from the party for a few minutes, to say how very glad I am that this symposium has been held, and also on behalf of myself personally, on behalf of the college, to say how pleased I am that we have a new chemistry department, a new chemistry undergraduate and postgraduate programs, a new chemistry research programs. How glad I am that we have chemistry. I'm not a chemist, as you might have guessed. I'm a historian. Indeed, I think this may have been the last paper, the first chemistry presentation I've heard in 48 years. The discipline's moved on, I'm happy to say. Seems to be in a very good condition. But even with that long personal separation from chemistry, it's self-evident that chemistry is a hugely important discipline. And after this unfortunate absence of a chemistry department from King's for about 10 years, a process which, reflecting a series of decisions taken before I became principal, and I'm happy to assure you. I'm delighted that this hole in the college's academic offerings has been plugged. And it was very gratifying to me also that when we began constructing the new department, that we were so successful in attracting very, very talented people to staff it. And I think Evelyn may have a word to say about the composition, the profile of that new department. I'll just say that though I wasn't personally involved in the recruitment, that all the reports that my colleagues brought back to me about the people we were recruiting were quite spectacular. And the people I've met since they joined us have certainly borne out those euphoric reports that I received in the early stages. I'm going to leave the thanks to the people at King's who have been directly responsible for the new chemistry department and the new chemistry programs to Evelyn. But I do want to express briefly very sincere thanks to two individuals, Phil Blower, of course, who's been the head of the department, and to Roger Morris, who, in addition to his duties as, which have been heavy ones, as head of our School of Biomedical Sciences, has taken the time to inject a lot of dynamism into the introduction of the new department. He has blazed the trail, and he and Phil deserve, along with many other people, a great deal of credit. I'm also glad that this symposium has attracted this large and distinguished audience. Some of you I know are from within King's. One of the paradoxes of the period when we didn't have a chemistry department is that, almost by definition, given that we had this very large medical school, we have a pharmacy, big and distinguished pharmacy program, all sorts of other things which are staffed in large measure by people with chemical training and whose teaching and research is in part chemical, like our Dental Institute and so on. Now we have a chemistry department and a series of chemistry programs that those chemists and people with chemical interests within King's College London can interact with in a serious way in directly chemical pursuits. But I'm also very pleased that many of you have come from outside King's, because clearly a new department and a new program. We're actually the fourth oldest university in England. I think I worked for 21 years in a medieval Scottish university, so we've got to be careful about this sort of thing. The fourth oldest English university, but however old a university may be, it needs friends, and that's true of each of its major programs. And I'm delighted that so many people of such distinction have come from outside King's, and that you're part of the community now that are interacting with and helping to sustain our new chemistry program. So thank you all. Thank you for listening to me. It's my pleasure to introduce my colleague, Professor Evelyn Welch. Now, she's not a chemist either, as she'll probably explain in a moment, but she is our Vice Principal for Arts and Sciences, and that includes our School of Natural and Mathematical Sciences. And even more to the point, she's taken an initiative in the few months that she's been here to launch an overall science strategy for King's, transcending the boundary between our arts and sciences schools, including natural and mathematical sciences, and our health schools, including the School of Biomedical Sciences. So she's well worth welcoming to this symposium. Please join me in welcoming her.
This is the really dangerous bit, when it really literally is only me between you and the drinks. I asked specifically Roger whether I could say something at the end of this event, because as a historian, like Rick, I wanted to remind all of us that chemistry has been part of King's lifeblood since its foundation in 1829, with its first foundation professor in 1831 of John Frederick Daniel, who was the professor of chemistry here. And for those who come to the Strand campus, you can actually see his electrochemical cell, the Daniel Constant cell, in our staff common room. And I really do recommend looking at this very, very beautiful bit of scientific apparatus there. And he was a typical King's professor for his time there. He worked across medicine, chemistry, meteorology, geology, and was elected a Fellow of the Royal Society at the age of 24 in 1814, like all our professors here at King's there. He also died while speaking at the Royal Society in the 1840s, perhaps not like all of our professors here at King's there. But equally, of course, and very memorably, although many people forget that she had her PhD in physical chemistry, Rosalind Franklin worked here with the biophysicists there. And it's that tradition of interdisciplinarity, moving across boundaries, research and teaching excellence, and about having an astonishing number of female scientists here at King's, which we've been able to renew with the formation, the reformation of the Department of Chemistry. Because even after 2003, although we didn't have quite as many chemists going down the undergraduate aisles at graduation, chemistry never left King's. And it's a real pleasure that we can put a plaque on the door and bring together the many friends of chemistry and the chemists themselves, and look forward to all of you thinking of yourselves as having an affiliation, an association, and who knows, maybe in the future, a position as an FRS and a professor at King's, and we'll try to make sure that you don't die while speaking at the Royal Society, as Daniel did. But before I finish, as Rick has said, the revitalisation of chemistry, the bringing it back together, has been due in no small part to a very large number of people, and above all to many of the new recruits who've come in this year there. But it's been very much Phil Blur and above all Roger Morris who've made this initiative happen. So in ending this day of celebration, and I'm sure it's the first of many toasts, could we say thanks to Phil and to Roger and to the entire chemistry team and to all of those who made this event happen. Thank you very much.